Thank you very much for that. And in beginning, I'd like to thank the Academy for this great honour of election, but of course reflect on the fact that this has really been a team effort for very many talented people in our group since 1998 who've worked with us as students, postdocs and RAs. I'd also like to acknowledge Jane Visveda, who's a Fellow of the Academy as well, long-term collaborator, and my wife. So this pie chart really tells you the story of the problem that we face in terms of breast cancer. It's by far the most common cancer to affect women, and even though the mortality figures have very substantially improved over the last couple of decades, there are still about 8 women dying per day from breast cancer, and it's estimated worldwide that at least 500,000 deaths occur from this disease. So the way that our group has been trying to tackle this problem is to really get a better handle on the cellular and genetic underpinnings of how breast cancer arises in terms of how things go awry from normal developmental biology. We're very keen to know how female hormones play a role in breast cancer development, and thereby apply the findings that have been made to better treatments or preventions for the disease. And much of this, in a sense, is underpinned by the belief that breast cancer is very much a caricature of normal development gone awry. So it really doesn't take that many genetic mishaps for normal development to be skewed towards proliferation that's uncontrolled. The cell types in the breast which are prone to cancer are these epithelial or ductal elements, which are of course very important for producing and delivering milk during lactation. So these are the key elements within breast tissue that we've been very interested in studying. And this shows one of those ducts, and you can see it's a very elegant bilayered setup whereby you have an inner layer of luminal cells, there you can see in blue, and the myepithelial cells on the outside, which provide the contractile properties to the ducts and essentially help them keep their shape. Of course, it's not quite as simple as that bilayered duct. When you actually drill down and look into the cell types that produce this duct, there are likely to be many distinctive cell types within the mammary gland. And of particular interest are the stem cells, or these blue cells here, that play a very important role in establishing these ducts during normal development and also maintaining them during normal physiology throughout life. So in one sense, stem cells can be considered the mother of all of this mammary epithelium. They give rise to daughter cells or progenitor cells, and then a succession of more mature cells along the way. And our interest is in classifying these, but also understanding the genetic mishaps that can happen to these cell types that can give rise to different types of cancers. The breast development's actually very interesting from a developmental point of view, because most of the action occurs postnatally. Really, at birth, there's just a rudiment present, and it's really the surge of female hormones through puberty or pregnancy which leads to the full developmental capabilities and changes within the breast. And the fact that mice and people can undergo many rounds of lactation really speaks to the need to have a repository of stem cells that will help reproduce these cells multiple times. So next I'd just like to show you a few sets of images which show you how dynamic these changes are. And on the left here you can see just the small unlager that's present at the end of embryonic development. And in the middle panel, really the gland in waiting pre-puberty. And in a mouse, at least over a 2-week period, there's a very profound growth and penetration of these ductal elements to fill the mammary fat pad through the delivery of a surge of female hormones. Anne Rios in the lab has been able to master 3D imaging in a way that she's able to look at these events in a native gland without any enzymatic digestion, which is giving us really deep understandings of these events at a molecular and morphologic level. With the onset of puberty, there are very profound changes, and these secretory units can form. And this Christmas tree effect that you can see here really represents different pockets of stem cells that have given rise to uniform patches of tissue as these secretory units develop during pregnancy. And the profound dynamic changes continue with lactation, and here you can see a fully mature gland that's really primed to deliver milk, which is obviously the full functional requirement of the breast. So I'd like to return to how these distinct epithelial cell types arise and what's the role stem cells play in producing these ductal elements. And about a decade ago now, Francois Vaillant and Marc Chappleton in our labs worked for many years to try to prospectively isolate stem cells, and were ultimately successful in identifying a population that could give rise to mammary outgrowths that had stem cell properties. And quite remarkably, even a single stem cell from this population was able to produce mammary outgrowths. Over the next decade, many people in our group have been able to define this epithelial hierarchy from the stem cell to the daughter progenitor cells and the mature cells. And we and other groups around the world have been able to prospectively isolate these cells and then study their characteristics. One of the most important, I think, interesting findings that was made early on was through the gene profiling of different subsets, and it became clear that the different types of breast cancers very much mimicked distinct cell types in the gland. For example, hormone receptor positive breast cancers looked much more like a mature luminal cell, whereas clinically aggressive so-called triple negative cancers looked much more like a luminal progenitor cell. And this is important because it allows us to, in a sense, have a shopping list to go out to look at how we could target these cells preemptively for prevention or in therapy. One of the populations of carriers that we looked at who are particularly prone to breast cancer are women who carry BRCA1 mutations. And the reason for looking at BRCA1 breast tissue is because women who harbour these, a single copy mutation in their genome, are at a very high risk of developing breast cancer, some 65% lifetime risk. And the tumours that arise are often highly clinically aggressive. And what was interesting to discover through the work of Elgene Lim and Francois Vaillant was that it appeared to be the luminal progenitor cell, which was the culprit cell which was misbehaving and likely to give rise to the basal-like tumours that arise in BRCA1 mutation carriers. I'd like to briefly turn to the exploration of how female hormones might impact on stem and progenitor function. And one of the things that Marie-Liesse Asselin-Labat in our lab did was to try to characterise which of these different cell types express female hormone receptors and would therefore be responsive to female hormone signalling. And what was quite surprising at first was that the stem cell compartment actually lacked hormone receptor expression. It was really only when you got towards the progenitor or mature cells where you started to see female hormone receptor expression. That was looked at through a variety of approaches to try to manipulate the system through the removal of hormones or addition of hormones. And what became quite apparent was that even though the stem cell lacks hormone receptors, it's very highly responsive to hormones in an indirect fashion. So it's possible by removing the ovaries or by some form of estrogen deprivation to switch off stem cell activity in the breast. And conversely, exogenous hormones or the hormones of pregnancy can reactivate stem cells and really boost their numbers and activity. And Marie-Liesse was able to identify, in collaboration with bioinformatician colleague Gordon Smythe, a molecule called rank ligand that has emerged as a very key paracrine regulator that helps this happen. So what is currently believed is that female hormones can activate a hormone sensor cell which has female hormone receptors, and these then in turn produce factors such as rank ligand, which can then feed back and switch on stem and progenitor cells in the breast. So this finding has fairly profound potential implications for breast cancer prevention, because one hypothesis would be that if you can switch off rank ligand signaling, then you will switch off the proliferation and growth of these precursor cells in the breast. And that's something that we're currently pursuing by looking at BRCA1 mutation carriers in a pilot study to understand whether that is indeed possible, that treatment with a rank ligand inhibitor could possibly do that. We're fortunate because there's actually an inhibitor in the clinic called denosumab, which could potentially be repurposed for this. Denosumab is currently in use to treat osteoporosis or breast cancer that's spread to the bone. So I'd like to finish and once again acknowledge many mentors that have got me here, very wonderful and talented, enthusiastic people who've worked in our laboratory over the years, and of course Jane, who's very much been my partner in crime for this work over more than 15 years now, and many clinical colleagues and patients who have also contributed to this work. Thank you.
Thank you, Jeff. Any questions? Yes. Could I have a...
I was very interested in the last part of your talk, because isn't rank ligand also involved in bone formation through the control of osteoclast signaling, and therefore if you actually got rid of it, wouldn't this have very deleterious effects on bone structure?
So that's a very important question, and I guess just taking it back a step, it's interesting to think that bone and breast have, in a sense, borrowed the same biochemical pathways for their operations. Milk production is a time where you need a lot of calcium, bones need a lot of calcium, and these pathways have been hijacked. You're correct to say that switching off bone turnover could have long-term deleterious effects, but denosumab is actually used to treat osteoporosis, and at low levels there seems to be sufficient turnover for that to be safe and effective. It's also used to treat breast cancer, and in fact recently there was a study by an Austrian clinical trials group that used relatively low doses over a 5-year period that appeared to be safe and tolerable. But I agree, when you're thinking about a prevention drug, you want something that's extremely safe, because you can't make a well patient necessarily unwell. It's really not their expectation. They really want to have an adverse event like cancer prevented, not a new adverse event.
Another question from Roger.
I enjoyed that presentation, and I think breast cancer is fascinating. To begin with, it's an occupational health disease of nuns, and the evidence is absolutely clear-cut. How would you suggest that we protect nuns from breast cancer? And as a supplement to that, in view of the excellent evidence which shows that pregnancy confers a long-term protective effect against breast cancer, what do you think that one pregnancy is doing to the cells in the breast for the rest of life?
Thanks for those insightful questions, Roger. I guess the first answer is to make sure that nuns go out and have as many children in their teenage years as possible, because it seems to be the early activation and functional maturation of the breast which makes a difference. So as you point out, parity protection is very powerful, so having children at a very young age is a powerful prevention strategy. But of course in our society we've seen quite the reverse with late pregnancies. It could be possible to explore the hormonal manipulation through, if you like, a pseudo-pregnancy, although that may not be particularly palatable in the clinic. But conceptually that would work very well. Agents such as rank ligand inhibition might be another way for nuns to pursue this question.
Don't you like it when people who ask a question answer the question themselves as well in advance? I think we'll call the questions to an end here, and thank you, Jeff.
Thank you.